Hello friends, welcome to my channel Biophil. So today I will complete the rest of the portion of fungi as I completed the general description of fungi in part 4a video, that means the previous video of this. So today I will discuss medical significance of fungi. So let's start the video. If you like this video, please like, share and comments and subscribe my channel Biophil and help me to grow. Let's come to the first slide. It's the introductory part of fungi, that means medical significance of fungi. So fungi represent a significant group of pathogens capable of causing a range of disease in humans. While the majority of fungi appears to be harmless to humans, it is worth bearing in mind that in the case of a profoundly immune compromised individual, a wide range of fungi can present as capable of inducing disease. That means there are such groups of pathogen, that means fungi, so they can easily do disease to human, but the majority in case of immunocompromised patient or weak immune system containing patients or humans, it has profound effect on health. So ideas of mycology and the types of mycosis. So the study of fungi is called mycology, and the diseases they cause is known as mycosis. So mycosis are classified depending on degree of tissue involvement and mode of entry into the host. So we will discuss the type of mycosis in next slides. First, we will discuss primary and opportunistic mycosis. So, the primary pathogenic fungi are able to establish infection in a normal host, whereas opportunistic pathogens require a compromised host in order to establish a function. Example: cancer, organ transplantation surgery and AIDS. So these are the such diseases or the condition where this opportunistic fungi can cause an infection in human beings. But the primary pathogenic fungi are normally, they can establish infection in normal host. Whether they are healthy or unhealthy, or immune system is strong or weak. Most cases of primary deep mycosis are asymptomatic and clinically mild infection occurring in normal patients living or traveling in endemic areas. That means primary deep mycosis, that means this primary pathogenic fungi, if they cause infection in human being, they must be sometime mostly they are it is asymptomatic or clinically it has some mild infection. It never show huge infection in normal patients, as because their immune system is active. The primary deep pathogens usually gain access to the host via respiratory tract, and the opportunistic fungi causing deep mycosis invade via the respiratory tract, alimentary tract or intravascular devices. That means when we encounter with air, this spores of fungi, they can enter to our respiratory tract, and if they somehow cause disease to us, this is obviously caused by this primary pathogenic fungi. But in case of opportunistic fungi, whenever they cause this deep mycosis, they must enter to a respiratory tract. That means they should enter to inner portion of this respiratory tract or alimentary tract or some devices during some pathological test or experiment, if it is entered to our body, this way can they enter to our body. Okay, so this is the brief concept of primary and opportunistic mycosis. So these are some examples of primary mycosis caused by Coccidioides immitis. Say the disease name is coccidiomycosis, and it is due to inhalation of spores and it causes pulmonary infection. See, it's written also where mostly it can be seen. Some other examples are also present in this slide. You can clearly see this. Let's move on to next slide. Again, this slide represents the opportunistic mycosis. These are such examples of opportunistic mycosis, that is candidiasis caused by Candida albicans and other Candida species. This is mainly a yeast in yeast. That means this Candida albicans is kind of yeast. So it causes endogenous infection, primary infection to mucosa, skin with secondary dissemination. These are rest other examples also included in opportunistic mycosis. You can follow these examples and the details of this infection. Let's move on to next slide. So the third type of mycosis is superficial mycosis. So it's mainly localized to epidermis layer to our skin. So hair, nails, these are the structure where the fungi can cause infection, but extend deeper to keratinized tissue. That means hair, nails, these are the most preferable places where they cause infection, but they can go far deeply. This is most common mycosis in human and are acquired from the environment infected humans or natural animal hosts. The fourth type of mycosis is subcutaneous, confined to dermis. That means the second layer of our skin. That means first layer is epidermis, that is known as superficial mycosis, and the second one is subcutaneous mycosis, that is confined to dermis layer. That is after that second layer of epidermis, after that the layer of epidermis. First is epidermis and second is dermis. Subcutaneous tissue or adjacent structures are the target of this microorganism. That means the fungal targets are subcutaneous tissue or adjacent structures. These are infections caused by a number of different fungi that arise from injury to the skin. That means it's mainly due to the injury to the skin. They usually involves in dermis, subcutaneous tissue and muscles. It can spread to these tissues and muscles too. The fungi commonly live saprophytically on through bushes, roses, or tree barks from which the wound can offer. That means as because the humans are normally dependent to plant material. That means we need to collect wooden material from plant tissues or plant like big trees and all. So during that collection, if somehow some cut or wound happen to a human skin, so this microorganism can transfer from this bushes, this barks, from they can transfer to human wound, and after that they can cause infection to human skin or under the skin. So this is the way of their methodology of infection. See, this slide represents some examples of subcutaneous mycosis and cutaneous mycosis. You can go through this slide and I think you will get some ideas and example along with some details. As it is included sporotrichosis, Sporothrix cichini is the microorganism, the causative agent of this. And cutaneous mycosis is mainly caused by dermatophytes. That means dermatomycosis are main pathogenic agent for this infection, like microsporum species, then trichophyton species. These all dermatophytes are filamentous fungi. They are zoophilic, that means their host range mainly are animals, that means they can, humans as a host and other than other animals are also there, secondary host or primary host, whatever. So they have two kinds of hosts. Also, they can transmit it by direct or indirect contact. They can easily transfer to human and cause disease. Let's move on to next slide. So, basic concepts of environmental epidemiology. So, the epidemiology of dimorphic primary pathogens may be contrasted with that opportunistic fungal pathogens. So, these four types of microorganism, as I mentioned before, they have some epidemiology, like as it is mentioned word, that is dimorphic. So in my previous video I already explained the dimorphism, this term, so you can go through this and I think you will get clear idea of this dimorphic term. So the primary pathogens have a relatively well defined geographic range of endemic infection in immunocompromised host. By comparison, opportunistic fungi like Aspergillus species are ubiquitously distributed, with the frequency of infection being dependent upon population of immunocompromised hosts. Penicillium marnifi is opportunistic pathogen, appears to be geographically restricted to East Asia, particularly Thailand and China. That means, see, primary pathogen, they can cause infection to normal hosts, whether their immune system is weak or strong. That doesn't matter. Okay, so that is the reason they have a large geographic range. That means they can present almost a bigger area, bigger geographic range they have, and they can also infect immunocompromised host along with normal host. But in case of opportunistic fungi, as they are totally dependent to immunocompromised host, but for their normal life cycle they are present in normal host, but that time they are not opportunistic. So they are only mutualistic to the host. But whenever their immune system somehow get weak, that time they can cause disease to them, or somehow some transplantation or drug entry, somehow if immune system get weakened, that time they can cause disease. That's why they are restricted to a specific geographical area. That means they have small geographical area in comparison to primary pathogens, that means primary fungal pathogen. Obviously, I think this is the whole epidemiology of this primary and opportunistic fungal pathogens. Let's move on to next slide. So these are the procedure by which we can diagnose fungal pathogen whenever humans get some infection, and after that microscopy, culturing method, serology, antigen detection, cutaneous test, nucleic acid detection. These are the tests by which we can diagnose a specific reason, that means specific etiological agent which is responsible for the specific infection to human. So move on to antifungal therapy. That means whenever fungal infection is there in skin or inside the skin or our deepest area of our body, there's such a therapy. That means some drugs are present. That means they are made. These are synthetic product which can somehow hamper the fungal cells. That means these causative agents. So the principal targets and mode of action of antifungal drugs are through disruption or inhibition of fungi. So this inhibition methods can be different, like these are Cell wall integrity, that means cell wall can be disrupted, cell wall biosynthesis can be stopped, RNA synthesis can be stopped, and cell division and nucleic acid biosynthesis. It is also a target of the antifungal drug. Let's move on to some example of antifungal drug. First type is polyenes. So this bind to sterile component of, that means ergosterol. As I mentioned in my previous video, ergosterol is the unique component of cell membrane of fungi. So this is the target of that antifungal drug, and they can hamper the structure of cell membrane. So example is nystatin. Next drug type is classification. These are the classification of different types of antifungal drugs. The second is azole. So these are synthetic compounds that can inhibit the ergosterol biosynthesis. First, it can bind to the sterile component, that means the ergosterol, and they can disrupt the structure by, they can difference the permeability, leakage, or this is the mode of action. Okay, but for azole, they can stop the biosynthesis, that means newly biosynthesis they can stop. Example is ketoconazole. And the third one is pyrimidine, synthetic analogs of pyrimidine. They can convert it inside the fungi to compound that replace the uracil of the RNA biosynthesis and interfere the protein production. See, this drug interferes with the RNA synthesis, and eventually it will hamper the protein production of fungi. Example is fluocytosine 5. Fluocytosine is the chemical name. Thank you so much for watching this video, and please comment how you are feeling after watching this video. Please subscribe the channel and help me to grow, and thank you so much for watching this video.